Hello, my name is Matthew Rocha, and today I will be going over the topic I chose, which was the topic on cloning. So, what is cloning? So, cloning is a technique scientists use to create exact genetic copies of living things. These things are such as cells, genes, tissues, and even animals. These clones are also made in a lab, and as you can see from the visual photo I put up, you can see the DNA in the sheep, and then it gets cloned into a DNA clone and a DNA or a clone of the sheep, and they're identical. So that's basically what cloning really is. So cloning genes. Scientists clone genes in order to better understand them and also to study them. In order to clone a gene, they take DNA from a living creature and put it into a carrier like bacteria or yeast. Also, every time a carrier reproduces, a new gene is made. And now going over the embryo twinning. So there are two ways animals are cloned. So the first way is embryo twinning. And what scientists do is split the embryo in half. Then both halves are placed in the mother's uterus. Now each part of the embryo develops a unique animal, and both the animals share the same genes. And I put a visual photo which exactly describes the process I just described. As you see, the embryo is divided into two halves, and then the surrogate mother gives birth to identical twins, which are the clones. So another way, the second way that I mentioned is the somatic cell nuclear transfer. So what are somatic cells, you may ask? Somatic cells are the cells that make up an organism but are not sperm or egg cells. So to make a clone, scientists transfer the DNA from an animal somatic cell into an egg cell that's nucleus and DNA have been removed from. The egg then develops an embryo that has the same genes as the cell donor. The embryo is then implanted into an adult's female uterus to grow. And as you can see in the visual photo I put, it describes the process. As you can see, this we have the somatic donor, the egg donor, and then we can see the nucleus removed from both donor cells, followed by the nuclear transfer, and then the cell growth and tissue culture, and then you can see it turns into embryo implantation into the surrogate cow mother, and then we got the clone. So Dolly the sheep. This is the first successful mammal to be cloned, and it was in 1996. And I believe it was Scottish scientists who were able to successfully clone a mammal. And she was cloned using a udder cell taken from an adult sheep. It took them about 276 attempts before actually getting it right. And since then, scientists have cloned cats, deers, horses, and even rabbits. Although we have not successfully cloned a human yet. And I know there's people who are against cloning humans due to how unethical it is, but it's interesting to see like where our advancements could take us. So yeah, that is it. So thank you for watching.